Hello, Assalamu alaikum. My name is Arif Ali Khan, Department of Microbiology and Field Seconds. My registration number is MB3 2022-2001. I am the student of Kohat University of Science and Technology, Kohat, Pakistan. So today I will present the topic of southern blotting, principle of southern blotting and its application. So what is southern blotting? Southern blotting is a laboratory technique used in molecular biology to detect specific DNA sequence. It is a classical technique which involves in separating of DNA fragment based on size by the use of electrophoresis and then transferring of them to a membrane hybridized with a labeled sequences specific probes, then washing them and finally detection of DNA bands. It's named after Edwin Southern, who developed the method in 1970. Principle. The southern blotting technique involves in several steps. The first step is about DNA digestion. The DNA sample is first treated with restriction enzyme to cut it into smaller fragments. The DNA which are extracted from different type of organism are first treated with restriction enzyme to cut into a smaller fragments here. The next step is gel electrophoresis. The DNA fragments are separated by a size using gel electrophoresis. The DNA is loaded into a wells which is present in gel electrophoresis and an electric current is applied here, resulting in separation of fragment based on size. The smaller size molecule faster toward opposite charge than a larger molecule because of molecular size. The next step is DNA denaturation. Here the DNA denaturation takes place. The DNA fragments are transferred from gel to a solid support, usually a nitrocellulose or nylon membrane, and here the blotting is takes place, applying pressure or electric field to transfer the DNA into this membrane or nylon membranes. DNA hybridization. In the nylon or nitrocellulose membrane treated with labeled DNA probes that is complementary to the target DNA sequence of interest. These membranes are then treated with labeled DNA probes which are complementary to the specific DNA sequence. The probe hybridized bind to the target DNA on the membrane, forming a stable DNA duplex. Detection. The label probe is detected using various methods, for example, autoradiography, fluorescent, chemiluminescence. This step allows visualization of specific DNA fragments that have hybridized with the specific complementary probes. Here the overall principle of southern blotting and its procedure. In first step, extraction and purification of DNA take place, which are extracted from different microorganisms or other organism cells. Then DNA is restricted with enzyme, generally a restriction enzyme used. Then separation by electrophoresis, denaturation of DNA, and transfer of these fragments into a nitrocellulose paper blottings. Add a label probe for hybridization to take place, washing of unbound bonds, and then autoradiography take place for visualization of the specific bands. Application. Here are some application of southern blotting. The first is about gene identification and characterization. In gene identification and characterization, southern blotting is very useful. Southern blotting is commonly used for identify and characterize specific gene or gene sequences. By hybridization label probe to the transferred DNA fragment, researcher can determine the presence or absence of a particular DNA sequence and study it by size or by numbers. The second application is about DNA fingerprinting. DNA fingerprinting is also useful by the use of southern blotting. Southern blotting can be employed in DNA fingerprinting or DNA profiling. The technique allows for the comparison of DNA sample to determine their relatedness or individual uniqueness. by using specific probe that target polymorphic region of the genome. In this method, researcher can create a unique pattern of bands for each individual, which can be used for identification purposes, forensic investigation, or paternity testing. The third application of Southern blotting is about detection of gene rearrangement and deletion. Southern blotting is useful for detecting gene rearrangements such as chromosomal translocation, inversion, or deletion. By probing genome DNA with specific gene probe, researchers can identify abnormality pattern that indicate structural alteration in the DNA sequence. The fourth application is about study of DNA methylation. Southern blotting is also useful in the study of DNA methylation. Southern blotting can be used to investigate DNA methylation pattern also, which will play a crucial role in genome regulation and epigenetic modification. By using methylation specific probe, researchers can identify the methylation state of specific DNA region, providing insight to gene expression and potential epigenetic modification associated with various diseases. The next application is about genetic disease diagnosis. So the Southern blotting is also useful and applicable in genetic disease diagnosis. Southern blotting in clinical diagnosis to study and identify specific mutation or alteration associated with inherited disorder by using specific probes which are disease specific. In this method, researcher can detect the presence of disease causing mutation or variation, aiding in accurate diagnostic and genetic counseling. So blotting have many applications due to their high sensitivity, faster turnaround time, and ability to analyze multiple targets. Thank you for watching.